lock and key hypothesis or theory. When you say a lock is a padlock, and then a key is the key which is used to open the padlock. You cannot bring another key and then it opens that padlock. No way. That's why we are saying that these enzymes, they are specific in action. When it is acting on this type of food, it cannot act on the other type of food. So we are saying that it is using what you call key and lock hypothesis. So where the key enters from, that small hole where the key enters from, we call it an active site. Then the key which enters into the lock, yes, we call it a substrate. That is the food this enzyme is going to break down, or is the food the enzyme is going to build up. So what happens, we call this one, the enzyme has a particular shape. It has a specific shape, each enzyme. And then this substrate, the substance on which enzyme work is called a substrate. So the food in which this enzyme is going to work on, then we call this food, which we call it substrate. We don't call it food, we call it substrate in science. And then the active site, this is the part of enzyme to which the substrate become attached. So this is where that small hole, that's what I've talked about, that small hole, so where the key is supposed to enter is what you call the active site. So basically, that's what you need to know about enzyme. You need to know how to label that. And then how does it work? The lock and key hypothesis, you start with the enzyme, and then on top you have what you call the substrate. Yes? Now this substrate is supposed to fit in this. If it is a different substrate, then it won't be able to fit. So this substrate comes and fit this one here, and then this one's there. And then what happens now, it fits in here. Yes, you see, it fits in here. The enzyme substrate product complex. So it forms a complex. Yeah, it has not yet broken it down, but it forms what you call a complex of enzyme and substrate. So now the key has entered, but it has not yet opened it. It has not yet opened the what? The lock. So what happens? So now the products are leaving the active site. It must open the lock, and then the key must be removed from the what? From the lock. That's how the enzyme works. Then what happens? The products leaving active site of the enzyme. So these are the products which leave the active site of the enzyme. After that, after being broken down here, then now they leave the enzyme. It means that now the products have been formed. What we wanted is you have this substrate, yes, this food, it must be broken down, but we're gonna use this enzyme so that now these substrates can be broken down. So now you see that now they are done being broken down. Now they are being freed, and then now this one is going to go back and it's going to be used up. Sorry, it's going to be used over and over and over again. You see that the enzyme did not change. It remained the way it was. It was just to break down this substrate. So basically, this is trying to explain what's happening. A substrate fits into active site. It fits into active site, forming B, an enzyme substrate complex. So form enzyme substrate complex, and then a chemical reaction occurs and the substrate change the shape. Then you're saying that D, the product form, the enzyme can now react with more of the substrate. So you're saying that the products, they form, and then the enzyme can now react with more of the substrate. It means that it goes back and then reacts again over and over and over and over again. So that's how this works. We love this. We love to ask this. We really love to ask you that. Then you're saying that what are some of the factors which affect the rate of the factors that affect the enzyme action, or the way how enzyme is supposed to work? Number one, temperature. We saw it, that if you increase the temperature, it denatures the enzyme. It means that the enzyme changes the shape. An example here is the eggs. You see, when the eggs, when you are going to fry them, when you put them on the frying pan, then it changes the shape. That is what called denaturation. It has changed the shape, and you cannot bring back the eggs the way they were. It has been permanently changed. That's how the enzyme is. And remember, eggs are made up of a biggest percentage of it is what? Is protein. So it also affects the enzyme. Nature and the amount of substrate. The enzyme will work faster if the substrate is too much. It will work faster if the substrate is small, like small grains. The surface area is big. Then it means that you work faster than when it is just a big substance. That's why when you are eating, you chew so that you break down those small, those big particles into smaller particles, which can be acted upon by these enzymes very fast. So the nature, how it is, small pieces or big pieces. Small pieces will be easier to be acted upon by enzymes than big substances. And then if there are many, then it means that the enzyme is going to act upon them very fast compared when they are few, because they are not easily be located. Then another one is the amount of enzymes. If the enzymes are many, then it means that the rate of reaction is also increased. But a time comes whereby even if you increase the number of enzymes, still the rate remains what? Remains the same. Like low pH, enzyme is gonna be denatured, and then the activity will what? Will stop. Optimum pH, we will have the maximum enzyme activity. It means that that is the best pH the enzyme it can work in. We call it optimum temperature, optimum pH. Yes. High pH is going to denature still the enzyme, and then the enzyme will what? Will stop, or the activity will stop. So here is an example. This is an enzyme, pepsin. This is amylase, and then this is trypsin. Pepsin works in acidic medium from 0 to 8 pH. This is the pH, this is the activity. You have find out that it is maximum when the pH is 2. If it is, now it goes to alkaline or to neutral, then the activity stops, you see. So it means that when the pH is acidic, then pepsin is going to work. And its optimum pH is 2. The pH of 2 is the best pH for this pepsin to work. We find this in the stomach. And then you have this one, it works in neutral. We call it amylase. Amylase works best in in neutral medium at a pH of 7. Yes. So below that, yes, it's gonna slow down until it reaches 0. And then above that pH, above 7, it's gonna slow down until it reaches what? It reaches 0. So when you change the pH, it will affect the rate at which this enzyme works. Then you have the trypsin. Trypsin works in alkaline medium, and the best pH is 8. So below that, it is gonna make it to work slow, and then until it reaches 0, or above it is gonna also do the same. It's gonna reach a point whereby it cannot work anymore. So that's how this graph is supposed to be interpreted. So best pH is 2, best pH is 7, best pH is 8 for those enzymes. So what happens if the pH goes wrong? Here you have the enzyme, it works best. So what is happening if you change the pH, for example, for this pepsin, and then you put 8, you put pepsin in 8, what will happen is going to denature that enzyme. And then this one, the substrate, cannot fit snugly. The key, you have destroyed the padlock, you have destroyed the lock. So it means that the key cannot enter. It cannot fit in the active site snugly. Then what happens? It means that this substrate cannot act upon. No, the substrate cannot be acted upon by the enzyme. If you look at the shape here and the shape here, this shape is different. So it means that the reaction will not take place. That's why you see that it is by 0. And then the temperature is still the same. If it is low, it will be inactive. And then the rate at which this enzyme is working is gonna be low because of the low energy. And then optimum temperature, that's the maximum activity it can take place. High temperature is gonna denature the enzyme, and then the action is going to stop. So it works best in optimum temperature and the optimum pH. Let's look at the graph of it. What happens at 0? The rate is very low, very low. But as you increase the temperature from 0, the rate increases. When you reach around 35 to 37, wow, it is best. That's the optimum temperature. Now, if you start to increase, then the temperature is going to, the rate at which this reaction is taking place is going to reduce until it reaches 0. When you reach 60, all the enzyme which is there has been denatured, and then the reaction cannot what? Cannot take place. So this is what I was trying to explain, that due to here inactive, the enzyme is inactive, but when it reaches here, the enzyme is maximum. Yes. And then above that, then the enzyme starts to drop. So you see that this is the kind of different kind of enzyme from this. Because here the optimum is around 37, and then here the optimum is around 41. So we have some enzymes which can work more. For example, those ones found in the thermobacteria, like those ones which found in hot springs, they can work in temperatures which are above normal. So what happens when the high temperature is there? You heat. When you heat the enzyme shape will change. When it changes, then it means that still the substrate cannot be acted upon, and then it will leave without being acted upon. So it means that the action will stop completely if you overheat, or if the temperature is above the optimum temperature. It keeps on reducing until when the whole process cannot take place. So basically, that's it. That is enzymes in everyday life. This one, we are saying that where do you use enzymes in everyday life? You find enzymes, they are used in meat tenderizing, like the skins, removal of hair from the skin. Those shoes you put on, they are nice. Sometimes as a result of, they just remove the hair, and then you look at a nice shoe, but you don't know that whether it's, you don't know that it just came from an animal, because they removed the what? The hair from the skin. Then another one is used in making beer, wine and vinegar. We're gonna look at this one in grade 11. That how do they use these enzymes to make beer, to make wine, to make vinegar? Yes. Then another one is they are also used to make chocolate. Chocolate and syrups. They're also used to make a washing powder. Actually, it's the enzymes which remove the stains in the cloth. Yes, they are enzymes. They remove the what? The stains in the cloth. I'm just trying to describe. Different enzymes are used. For example, the proteases, then it can remove the blood, eggs, and gravy. Then you have amylase. This one can remove starches. If the stain is starch stain, then amylase will be used in that regard. That's why you find that they have different colors. Yes. Then lipase will remove the fats and grease. So all of these enzymes are added in the washing powder, so that if is there any protein in the cloth, it can be removed. Is there any starch in the cloth? It can be removed. Is there any fat in the cloth? Yes, it can be removed.